Hello everyone. So we will continue this chapter about the chromosome theory of inheritance. So last time we reviewed sex determination and mitotic cell division. So today we will continue with meiosis. So meiosis is a special nuclear division in which it occurs only in special types of cells. These cells are called the germ cells. These germ cells actually they are the precursors to gametes. So these are the mother cells that they will produce the gametes, the functional sperms and oocytes. So these cells, because they are regulated differently than the cells that they will produce somatic cells, so usually during development, during embryogenesis, they stay away from the other types of cells and they are regulated in different way. Once the reproductive organs are formed, these germ cells they will migrate toward the reproductive organs, and they will reside there. So if we review what happens during meiosis, so this cell has a diploid number equal to four chromosomes, and similar to what happens in mitosis, these cells they have to go through the cell cycle, so they have to replicate their DNA. So after they replicate their DNA, now the chromosomes are represented by two sister chromatids, but instead of doing one round of cell divisions, meiosis, uh, two rounds of cell divisions occur, in which in the first round the number of the chromosomes will be reduced into half. So we started with four chromosomes. We end up at the first round with two cells, each has two chromosomes, but notice that these chromosomes are represented by two sister chromatids. So during meiosis 1, homologous chromosomes get separated while the sister chromatids remain attached to each other. And during the second stage of meiosis, now sister chromatids split, so we end up with four haploid cells. Each one has half number of the chromosome. So we started with 2n, now we have four cells, each has 1n, half number of the chromosomes. In the zygote stage, now is dissolved, so there is no synaptonemal complex, but these homologous chromosomes cannot be separated from each other. Why? Because the sites of exchange, which is called the chiasmata, still connects the non-sister chromatids with each other. So these chromosomes, while moving toward the metaphase plate later on in the cycle, they will be moved as tetrads, as four strands of chromatids. This stage, the diplotin stage, usually in mammalian meiosis, oocytes are rest at this stage until the oocyte ovulates. So sometimes this takes 30 to 40 years to happen. The last stage is the diakinesis, and in this stage, nuclear envelope, which we cannot see in the previous stages, but at this stage, the diplotin stage, the nuclear envelope remains intact. At the diakinesis, nuclear envelope dissolves, and more condensation of the chromosomes. The next stage, so all of these substages, they were part of the prophase 1. Now in metaphase 1, the homologous pairs as tetrads, they move to the metaphase plate, and now you can see that they are connected to the spindle fiber. Remember from mitosis that kinetochore complex protein forms during the prometaphase in mitosis and in end of prophase in prophase 1 in meiosis. And in this case, the kinetochore complex will form one kinetochore complex per one chromosome. So per a pair of a chromosome, two kinetochore protein complex will form. And the main function of this kinetochore protein is to connect the chromosome to one spindle fall. And the same thing for this pair of a chromosome. So only one kinetochore complex will form here, and another kinetochore complex will form there. During anaphase 1 now, remember what keeps the sister chromatids attached to each other, a protein called cohesin complex. And what keeps these homologous pair connected to each other is actually the chiasmata, the sites of exchange between the non-sister chromatids. So for this pair to split, this homologous pair to split, separase, the enzyme, separase gets activated, and this enzyme now will cleave the cohesin bonds, the cohesin proteins that is at the tip of the chromosome. We call it the telomeric cohesin. So separase gets activated at anaphase 1 to cleave or to remove the telomeric cohesin. By cleaving the telomeric cohesin, now these chromosomes, these homologous chromosomes, can move away from each other. And you can notice that each chromosome now is a mix between two colors, the dark red and the light red, the dark green and the light green. Why? Because of exchange of non-sister chromatids of the homologous chromosome. So now we cannot call these chromatids as sister chromatids because they are not completely similar or identical. We call them recombinant chromatids. By the end of meiosis 1, telophase occurs, so opposite to what happens during the prophase, in which chromosomes decondense, the nuclear envelope will reform, and cytokinesis occurs, split of cytoplasm to split the cells into two. But instead of these cells of going to a round of cell cycle, they will arrest for short period. It's called interkinesis, so a stage between meiosis 1 and meiosis 2, which does not involve DNA replication. Instead, the cells may synthesize a little bit of a protein, relocalize messenger RNA, and then they will start the second stage of meiosis. It's called meiosis 2, which is very similar to what happens during mitosis. So in a prophase 2, chromosomes condense again, centrosomes, they start moving away from each other, microtubules polymerize, kinetochore now they will form, but two kinetochore complexes will form per one chromosome. Per one sister chromatids or recombinant chromatids, one kinetochore complex will form. And now the chromosomes will be arranged at the metaphase plate as single chromosome, not as pairs of chromosomes like what happened during metaphase 1. Now separase gets activated again in anaphase 2, but to cleave the centromeric cohesin, the cohesin that is only present at the centromeric part of the chromosome. Why? Because the telomeric cohesin is already cleaved. It's gone previously in anaphase 1. Now these recombinant chromosomes are free to move, and they will move toward viewing meiosis and mitosis is to recognize the differences between these two stages. One of them is what happens during a prophase 1 of meiosis, most importantly, the crossing over the stages of meiosis 1. Very important to see the difference between meiosis 1 and meiosis 2. In meiosis 1, the homologous pair will be arranged at the metaphase plate, while in metaphase 2, chromosomes will be arranged as single chromosomes. Um, in anaphase 1, telomeric cohesion gets cleaved, while in meiosis 2, centromeric cohesion gets cleaved. The diplotin stage, as we said, um, synaptonemal complex resolved, but still the homologous pair is connected by the chiasmata. So these are actually the sites that we can actually observe them microscopically, and we can see that chromosomes are represented by tetrads for strands and connected with each other through these sites. We call them the chiasmata. So sometimes error occurs during chromosomal segregation, in which either the homologous pairs are not separated from each other during meiosis 1, or sister chromatids remain attached and they are moved to one pole instead of getting separated. So we call this non-disjunctions, which is mistakes in a chromosome segregation during meiosis 1 or meiosis 2. So what is the result? What is the problem if non-disjunction happens? So we will end up with gametes with uh, increased number or extra number of chromosomes, while other gametes will have one chromosome less. And if this happens and these gametes are involved in uh, fertilization, so we end up with syndromes like, or chromosomal abnormalities like trisomy 21, which is called the Down syndrome, or Klinefelter, or Turner syndrome. So there are many chromosomal abnormalities that can be resulted because of non-disjunction errors during chromosomal segregation. And all of these syndromes, they cause phenotypes, abnormalities. Uh, you are familiar with Down syndrome, so the main characteristics of the Down syndrome individuals, they have problems in learning disabilities, they are sterile, they have enlarged heart and spleen, so they usually die earlier than expected. And here is a review between, oh no, here now, something to be convinced that the chromosome theory of inheritance is actually correct, that the factors that Mendel described is actually carried on this chromosome, is that when we follow what happens to the chromosome. So Mendel concluded that the 5 characters that he studied are actually controlled by genes, are located uh, on different chromosomes, or they were independently assorted. And if we follow what happens to the alleles, the different alleles of 2 genes, A and B, so, and we call this the independent assortment of a chromosome. See, so these chromosomes in green and red are homologous chromosomes. So during metaphase 1, they can be arranged in this way, in which the dominant allele of A and B will be split to one side, while the recessive allele of A and B will split to another side. Or the other orientation, or the other possibility, in which the dominant allele of A will be accompanied by recessive allele of B, and this will result with another 2 types of gametes. So if we have a diploid cell represented by 2 pairs of chromosomes, so, and the alleles of each gene is different. In this case, the cell is double heterozygous. We end up with 4 possibilities in which 4 types of gametes are produced: A capital, B capital, A small, B small, and in the other possibility, or in the other orientation, A capital allele with B small allele, and A small allele with B capital allele. Another way how genetic variation can occur is by recombination. So if genes C and D are located on the same chromosome instead of these genes are located in different chromosomes. So another way to recombine the types of alleles for these two genes is by crossing over, and which happens during prophase, during the packaging stage of a prophase 1. And now instead of the parental genotypes are C capital D small, C small D capital, we can get C capital D capital and C small D small, all types of different recombination. So think about sexually reproducing organisms. Think about a human. Why two individuals are not genetically identical, even if they are siblings, Juan or Khawat. One of them is because of independent assortment of these chromosomes, but in humans we don't have only two pairs of chromosomes, we have 23 pairs of chromosomes. So these 23 pairs of chromosome will produce 2 to the power 23 different types of gametes, and this is equal to around 8 million types of gametes. So in spermatogenesis, if all the genes in the father and the mother are different from each other, we expect to have around 8 million different types of gametes. So this is the maximum number of different types of gametes can be produced by the father, and another 8 million types of gametes can be produced by the mother, and by random fertilization these will produce around 64 trillion different types of gametes, because 8 million multiplied by 8 million, they will produce around 64 trillion types of different individuals. And this is a review between meiosis and mitosis. You can read the table and you can go back to the specific stages of mitosis and meiosis and see where is the differences. And now we will talk about gametogenesis. So meiosis is only one part of gametogenesis. It's actually the nuclear split of the chromosomes, while gametogenesis is more general, which is involve meiosis and involve other processes before and after the new and these, as we said, they are produced in special types of cells. It's called the germ cells. And in females, so here you have to focus both on this part of the figure, which describes what happens only to the chromosome, while the figure below represent other stages other than the split of the chromosomes, which more talks about the hormonal regulation and the other types of cells. So in females, there are a type of germ cells. They are reside in the reproductive organ. We call them the oogonia. And these oogonia, during early life of females, when the females are really infants, when they are really not born yet, these oogonia, they start to replicate by mitosis to produce large number of cells. These cells, after several rounds of mitosis, they will enter meiosis, and we call these cells the primary oocytes. So these cells, after they enter meiosis, they don't go far away, but they are rest in a stage in the prophase 1. Remember, we called it the diplotene stage, and these cells will remain arrested there. These oocytes will remain arrested there until ovulation. So just go back and remember that oogenesis starts really early on during female development when the females are infants and they do rounds of mitosis. These cells, they enter meiosis and they arrest in the diplotene stage of meiosis, and they will be remain arrested until ovulation, and ovulation occurs at adulthood. So adulthood around 15-16 years old, when the female matured, they start ovulating. So these cells, they are remain arrested there from early life until their time of ovulation. So when the female mature, some of these primary arrested oocytes, they will continue meiosis. So they will continue meiosis 1, and they will produce two types of cells. These two cells are haploid because in meiosis 1, chromosomes are reduced. But you can see here that one of these cells, they will have most of the cytoplasm. We call it the secondary oocytes, while the other cell has a little bit of cytoplasm, small amount of cytoplasm, and we call this cell the first polar body. So what happens during cytokinesis of meiosis 1 is unequal split of the cytoplasm between the two daughter cells. One of them will take most of the cytoplasm, while the other cell take only part of the cytoplasm. Only this one, the secondary oocyte, usually continue to meiosis 2, and again, unequal cytokinesis occurs to produce one mature ovum and a secondary polar body. Usually, the first polar body will continue without entering the second stage of meiosis. That's why we can see that the chromosomes remain as two sister chromatids instead of what happens to the secondary oocytes, in which the recombinant chromosomes or the two sister chromatids split from each other. Now, the cell that is formed by oogenesis is the mature ovum. Only one of them is capable of fertilization. The second thing that is important to notice here, that so the first arresting point during oogenesis, this is the first arresting point, occurs during the diplotene stage of meiosis 1, but there is actually a second arresting point. It's not drawn here, but these secondary oocytes, when they are ovulating, so secondary oocytes are the cells that are produced by the end of meiosis 1. They will enter a short period of interkinesis, then they continue to meiosis 2. So during the metaphase 2 of meiosis, they will arrest again. So during meiosis 2, remember that the chromosomes are still represented by two chromatids, and these cells will arrest at this stage. Do you know until when? Until fertilization. So if fertilization happens, this is the trigger for these cells to continue meiosis, in which the two sister chromatids separates. If fertilization does not occur, the cells will not finish meiosis 2. All right. So let's look at the second part of the figure, which represent not only the oogonia, these germ-line cells, the cells that are the mothers of the mature ovums, but also to the other parts of regulation. So as I said, these germ-line cells, or the oogonia, they reside in the ovary, the reproductive organ on the female. Females, they have two ovaries. So each month, upon hormonal regulation of hormone like the follicle stimulating hormone and the luteinizing hormone, one of the cells that are arrested in the primary oocytes, they will get activated. So what will happen to this cell, the primary oocytes? They start to grow by the induction of the follicle stimulating hormone. But these cells are not alone. They are surrounded by other types of somatic cells. So the sum of the germ cell or the primary oocyte surrounded by the somatic cells, we call it the follicle. So the follicle starts to grow, and these are the stages of the developing follicle. And at this stage, when the chromosomes are split, we can see that the secondary oocytes, they reach optimal size. They have grown until they can be released from the ovary toward the oviduct. So this released oocytes, these secondary oocytes are actually oocytes still arrested at the second stage of arrest, which is in the metaphase 2. And these oocytes, they move through the oviduct. If they encounter a sperm, then the meiosis 2 will be finished. If not, what happens during the menstrual cycle, the oocytes will be dissolved, and all the remaining part of the follicle, which is called the corpus luteum, will be released. So this is what happens during every month in the female period. So let's compare what happens during spermatogenesis. Starting from the time in which spermatogenesis occurs. So in females, we said that oogenesis starts really early in females when the females are infants, while spermatogenesis occurs in males when the male mature. And it occurs in the reproductive organs, in the testicles, and inside the testicles there are loops of cells or structures. We call them the seminiferous tubules, and these tubules, they have the spermatogonia, the mothers of the sperms. So during the spermatogonia, these spermatogonia will go rounds of mitosis to produce primary spermatocytes. But these spermatocytes will not arrest in the prophase 1 of meiosis, different than what happens in females. So they will continue. And here they will split by meiosis 1 to produce two cells. We call them the secondary spermatocytes. And these secondary spermatocytes, they are haploid. They have half number of a chromosome. In a human, they will have 23 chromosomes. And again, they will continue to meiosis 2 to produce 4 spermatids. But these 4 spermatids now, the haploid 4 spermatids, they are not ready to fertilize. They are not mature enough. Instead of the mature ovum, the result of female meiosis, these spermatids, they need to mature. They need to differentiate. So they enter a second stage of differentiation. It's called spermiogenesis, in which each one of these cells will form the typical shape of a sperm, the tadpole shape, in which it's formed from a head, neck, which contains the mitochondria, and the tail that can help the sperm to move. So big differences between spermatogenesis and oogenesis is the timing of in which these cells start uh, doing meiosis. So in males at adulthood, in females early infancy stage. Uh, the result of the spermatogenesis for mature or for haploid cells after maturation, they are able to fertilize, they are competent, while in females only one mature egg is formed, but by the end of meiosis, these cells are ready for fertilization. They don't have to do a maturation step. In spermatogenesis, cytokinesis is equal, so the size of the haploid product is equal, while in females they are in different size. In females, polar body they formed, and usually polar body they dissolve, they die, they do apoptosis, so they are not functional cells. While in sperms, all the haploid products will maturate and can be involved in fertilization. The end of spermatogenesis, and I will record another lecture maybe today or tomorrow about the validation of a chromosome theory of inheritance. See you next time.